Bismillahirrahmanirrahim. I am back with another educational video. This video is about the synapse and transmission of signals across the synapse. Let's begin. A synapse is defined as the specialized area of communication between two excitable cells. In nervous system, a synapse allows a neuron to send signals to another neuron. The term presynaptic cell refers to a cell that carries a nerve impulse towards a synapse. It is usually a neuron called a presynaptic neuron. A postsynaptic cell is the cell that carries a nerve impulse away from a synapse. It may be a postsynaptic neuron or an effector cell like muscle or gland that responds to the impulse at the synapse. The anatomical classification of synapses in nervous system is based on the type of cellular structures serving as the pre- and postsynaptic components. Most synapses between neurons are exodendritic, while others are exosomatic or they may be exoaxonal. Physiologically, two types of synapses, namely electrical and chemical, are recognized in the nervous system. It should be noted that electrical synapses are relatively less common than the chemical synapses in the mammalian nervous system. In an electrical synapse, there is a direct connection between two cells so that the ions can pass directly from one cell to the next. If one cell is depolarized in an electrical synapse, the joined cell also depolarizes because the ions pass between the cells. Chemical synapses involve the transmission of chemical information across the synapse. In a chemical synapse, a chemical signal called the neurotransmitter is released from presynaptic cell and it affects the postsynaptic cell. At an electrical synapse, action potentials conduct directly between the plasma membranes of adjacent neurons through gap junctions. The structure of a gap junction contains a central channel which acts as a low resistance bridge for directly connecting the cytosol of the two cells. As ions flow from one cell to the next through the gap junctions, the electrical potential spreads from one cell to the other. Gap junctions are common in visceral smooth muscle and the cardiac muscle. They also occur in the brain in few places. Electrical synapses have two main advantages. Number one, faster communication. Because action potentials conduct directly through gap junctions, electrical synapses are faster than chemical synapses. The events that occur at a chemical synapse take some time and delay communication slightly. The second benefit of the electrical synapses is a synchronous response. Electrical synapses can synchronize or coordinate the activity of a group of neurons or muscle fibers. In other words, a large number of neurons or muscle fibers can produce action potential in unison if they are connected by gap junctions. The value of synchronized action potentials in the heart is coordinated contraction of ventricular muscles to produce a heartbeat, or in the visceral smooth muscle to move through move food through the digestive canal. At chemical synapses, the presynaptic and postsynaptic cells are separated by fluid-filled gaps called synaptic clefts. The presynaptic terminal contains synaptic vesicles that are filled with several thousand molecules of a specific chemical substance, the neurotransmitter. The vesicles containing the neurotransmitter are aggregated near a specialized area of presynaptic membrane called the active zone. Neurotransmitter, when released, bind to receptors in the plasma membrane of the postsynaptic cell. Binding of the neurotransmitter to its receptor results in opening or closing of ligand-gated ion channels on the postsynaptic membrane. Arrival of action potential at presynaptic membrane is the first event in the release of the neurotransmitter. The resulting depolarizing membrane potential activates voltage-gated calcium ion channels in the presynaptic endings. These activated channels open and allow influx of calcium ions down their concentration gradient. The concentration of calcium ion increases inside the presynaptic terminal. The calcium ions associate with calcium binding proteins in the outer surface of vesicles containing the neurotransmitter. Activation of calcium binding proteins like calmodulin acts as a signal for merging of the vesicle with the presynaptic membrane. Next, the neurotransmitter is released through exocytosis into the synaptic cleft. Neurotransmitter molecules travel across the synaptic cleft and attaches with the receptor on the postsynaptic membrane. Postsynaptic receptors on the chemical synapses have been categorized into two varieties, namely inotropic and metabotropic receptors. An inotropic receptor has a channel that opens when the neurotransmitter binds to it. Ion flux occurs through open channels. Ion flux produces a graded potential on the postsynaptic membrane. These graded potentials act as electrical signals over a short distance on the postsynaptic cell. A metabotropic receptor is a complex that causes metabolic changes in the cell when the neurotransmitter binds to it. After binding, the G protein hydrolyzes ATP and moves to the effector protein. When the G protein contacts the effector protein, the latter is activated. In the case shown, the effector protein then acts on ATP to generate a second messenger, namely cyclic AMP. The second messenger can then go on to cause changes in the postsynaptic cell, such as opening or closing ion channels, metabolic changes, and changes in gene transcription. To summarize, this classification is based on whether the neurotransmitter binding site and the ion channel are components of the same protein or are components of different proteins. Chemical synapses are either excitatory or inhibitory, depending on how they affect the membrane potential of the postsynaptic neuron. When a neuron when a, when a neurotransmitter binds to the correct neurotransmitter receptor, an ion channel opens and a postsynaptic potential, either depolarizing or hyperpolarizing, forms in the membrane of the postsynaptic cell. A neurotransmitter that causes depolarization of the postsynaptic membrane is excitatory because it brings the membrane closer to the threshold. A depolarizing postsynaptic potential is called an excitatory postsynaptic potential. Although a single EPSP normally does not initiate an action potential, the postsynaptic cell does become more excitable. Because it is partially depolarized, it is more likely to reach threshold when the next EPSP occurs. A neurotransmitter that causes hyperpolarization of the postsynaptic membrane is inhibitory. During hyperpolarization, generation of an action potential is more difficult than usual because the membrane potential becomes more negative inside and thus even farther from threshold than on its resting state. A hyperpolarizing postsynaptic potential is termed an inhibitory postsynaptic potential. It is emphasized here that both EPSP and IPSP are examples of graded potentials. I recommend you to watch our video on the graded potentials. The link of that video is available in the description. A single postsynaptic neuron receives input from many presynaptic neurons, some of which produce EPSPs and some of which produce IPSPs. The sum of all the EPSPs and IPSPs at any given time determines the effect on the postsynaptic neuron. If the net summation of EPSPs and IPSPs is a depolarization that reaches the threshold, then a nerve impulse will occur at the trigger zone of a postsynaptic neuron. Now let's do a side-by-side comparison of contrasting features of electrical synapses with chemical synapses. In electrical synapse, cell membranes of adjacent cells are closely approximated. In chemical synapse, cell membranes of cells participating in a synapse are separated by the synaptic cleft. Gap junctions join cells in the electrical synapse, but no gap junctions are present in the chemical synapse. In electrical synapse, action potential is directly transferred from one cell to another. In chemical synapse, chemical messenger called neurotransmitter transfers signal from presynaptic to postsynaptic cell. In chemical synapse, transfer of signal from presynaptic to postsynaptic cell accounts for the synaptic delay. In electrical synapse, excitation is instantaneous without any delay. In electrical synapse, any cell can excite adjacent cell depending upon origin of the stimulus. In chemical synapse, a presynaptic cell will always excite a postsynaptic cell and not the vice versa. Thanks for watching and listening. Do share this video with your friends and colleagues. If you like this video, subscribe to this channel for watching more videos.